Good afternoon. My name is Hari Upadhyay, and I, along with Noel, are going to chair this session. We have 4 speakers, and each one with 30 minutes. And the suggestion is that we use first 25 minutes for presentation and 5 minutes for discussion. And when you have completed 24 minutes of your presentation, one of us will get up to indicate that only 1 minute is left, and we would expect that you stop your presentation. So without much ado, let me invite Andreas Graner to present high-density genotyping, phenotyping data, challenges of leveraging novel technologies for the valorization of plant genetic resources. Andreas, the floor is yours.
Thank you, Hari, for the introduction, and also many thanks to the organizers, in particular Varshni, for inviting me to this conference, where I'm going to share with you some of our results and considerations and thoughts about high-density genotyping and phenotyping. Although I know this meeting is mainly on genotyping, I take the liberty to also address the issue of phenotyping. Well, there are 2 major areas which we have seen developing over the past decade or so. And this is, as the title says already, and as we are aware of, this is the genotyping area. Next generation sequencing, but of course genotyping does not make any sense, or in many cases doesn't make too much sense if we do not connect the genotypes to the phenotypes, and therefore I think phenotypic analysis is something that must come along with phenotyping. Well, and also before I get into the topic, I would take the opportunity already in the start to acknowledge my co-authors, Jens Heilwagen, Christian Lukas, Uwe Scholz, and Martin Mascher are bioinformaticists, and the remaining people, Nils Stein, Joel Kuhn, Benjamin Kilian, and Kerstin Neumann, are the geneticists who basically complement bioinformatics these days. Right. If we talk about genetic resources, I think we more or less proudly can acknowledge that the mission of conservation of the genetic resources of crop plants has by and large been accomplished. There are more than 7 million accessions in gene banks. Worldwide, there are more than 1,500 gene banks with substantial holdings where those gene banks are located and what their size is. And also, in addition to just have the mere holdings, access and benefit to those genetic resources has been regulated over the past 15 years, mainly by the FAO in generating and drafting the International Treaty on Plant Genetic Resources, which clears or identifies and clarifies the regulations for access as well as the benefit sharing on genetic resources. And there is also an activity that has been founded or started maybe 6 years ago, approximately 6 years ago, and this is the Global Seed Vault in Spitsbergen, which is somewhere in the Arctic, where a repository has been established to have a safety duplication of those gene banks and those genetic resources. Up to now, some 10%, however, only of the genetic resources have made their way into the Svalbard seed vault, and there is certainly much room left for many of the major gene banks in the world to put their genetic resources and to safeguard their genetic resources in this sequel. Well, the conservation, the ex situ conservation of genetic resources basically is a reflection of the agronomic value of crop plants, and you find a clear correlation between the agronomic significance of a crop species and the number of accessions that are kept ex situ. And one of the most important crop species, as you know, or the more important crop species worldwide is barley, and therefore barley also represents one of the major collections, worldwide collections, in terms of ex situ conservation. Altogether, we have, or we find more than 450,000 accessions, barley accessions in gene banks worldwide, and the biggest collections have been listed here. And one of those major collections of barley we find at IPK-Gatersleben, our institute, which holds a collection that presently amounts to some 22,000 accessions. Right. However, to valorize the genetic resources, to make use of them and to apply them for plant breeding, we need to tag and attach information to those genetic resources and accessions, and one of the sources of information which we use is of course genetics, and if we talk about genetics, it's sequencing. And some of you might know that the barley genome is a fairly large genome. It's twice the size of the human genome, 5.1 gigabase pairs, and therefore it has been quite difficult to deal with. However, some 10 years ago, 8 years ago in 2006, the International Barley Sequencing Consortium has been founded, and since then major progress has been made regarding the genomic sequence of the 7 barley chromosomes, which are the base complement of all TBC species. And as of December 2012, when a milestone was reached, publication of the results has appeared in Nature, we had basically a physical back-contact map available that covers more than 80% of those 5.1 gigabase pairs. And this physical map was complemented and complemented by back sequences, by whole genome shotgun context, by markers that were anchored both to the physical map as well as the genetic map. And finally, this gene complement, or the gene complement of barley, could be determined based on RNA-seq and full-length cDNAs, and a total of about 27,000 high confidence genes were determined by 2012, of which some 60% were anchored to the genetic and or the physical map. Well, the consortium did not rest on its laurels, and it continued the activities towards the development of a barley reference map, and this mainly included, or also included, whole genome shotgun sequencing of a segregating population, which provided a wealth of additional marker data, in particular SNP data, along with sequence context that again could be integrated into this reference sequence or growing reference sequence. And here you just see the progress that we have seen over the past year in 2013 compared to the previous slide, 2012. And just to pick out a few of those figures, the number of anchored sequence contexts could be increased from 138,000 to more than 740,000 sequence contexts, whole genome shotgun sequence contexts that are now anchored to the genetic and that also means that the total size in megabase pairs of the anchored conflicts basically tripled from 410 megabase pairs to 1.2 gigabase. Well, and even more importantly, the number of anchored high confidence or high quality genes increased from 14,900 in 2012 to some 21,000 in 2013. Well, that's not yet the end of the game. As I mentioned, we are going and striving for a reference map, which is, I think, fairly important, in particular if you think of resequence exercises and activities. And the reference map is presently under construction. It is based on resequencing a minimum tiling path of backgrounds along the 7 Barley chromosomes. And here you see those countries that are contributing to this effort. And we expect that this minimum tiling path will be finished by the end of this year, by the end of 2014. So that certainly will be a major step towards the availability of a high-quality reference sequence of the Barley genome. Well, what to use this sequence for? One of the major applications we are interested in as geneticists is, of course, map-based gene isolation. And those of you in the audience who have been involved in the map-based cloning of genes, in particular in a complex genome, I'm just thinking of corn, looking at Roberto Tuberosa, or people who have been dealing with this challenge in wheat. They know that this has been a cumbersome work that frequently took, Frank is also sitting here, that takes more than took more than a decade to come down to the gene. And one of the bottlenecks of this exercise was the establishment of a back contact, physical contact, that spanned the targeting of interest in between the 2 flanking markers. I'm also looking at Andreas, who was the first actually to clone it. Well, right, so what we did here is basically the validation of the physical map, and the validation was performed in a way that we basically took the genes from the literature that had been cloned by the end of 2012 using a map-based cloning approach, essentially a map-based cloning approach. And then we were asking, do we find from our physical map back contexts that would cover those already cloned genes or loci of interest? And what you can see here in red are the contexts that we in silico, could just retract from the data of our physical map. And in almost all cases, those were fairly large contexts, frequently exceeding much more than, or even more than 10 megabase. And those contexts were usually much larger than in blue, the original contexts that were generated by this cumbersome approach to identify overlapping backgrounds manually from those libraries. So there is a huge, I think, benefit of the sequence for map-based cloning approaches, and the way the resources that we now have available in Barley really put the turbocharger on that. It's just illustrated by this small project here, map-based cloning of a multi-noded dwarf mutant gene or mutant allele. That was basically, or much of what was basically performed in the context of our master thesis. That means the whole project just took somewhat more than, let's say, 18 months or 16 months. Right, so we started with analyzing the cross between a wild type, this is Barke, and the mutant shown here on the left, and the mutant shows basically twice the number of leads or nodes than the wild type. So it's a morphological mutant that's easy to identify. Well, Then we had a population, and a two population comprising about 100 individuals. And the first exercise for a master's student is to make a chi-square test to see whether this is a monogenic segregation. And surprise, this fitted the monogenic segregation, which made him happy and us as well. And so we started a bulk segregant analysis by exome capture resequencing, and I think that was illustrated by Andreas already. So just to summarize what you see here, this is the bulk of the mutants, of the recessive mutants, and this is a bulk of DNA of the right type plants. And in case a gene is not genetically linked to the target gene, which would be the mutant gene, we would expect allele frequency. Here you see the alleles from this resequencing along the separate body chromosomes. You would expect an allele frequency of on average 50%, and that's indicated for both groups here by this line, 50%. And what you see immediately, and I marked chromosome 5 here, is that here in this area we see in both pools a clear deviation. So in the mutant pools there is a series of genes that has a frequency of for one allele of 100%, and that basically means those SNPs must, or many of those must be genetically linked to the mutant locus. And as a matter of fact, that turned out to be true when we did the individual analysis in the segregating population. And here in the top panel, you just see the result of the mapping exercise. So a series of SNPs, or the corresponding sequence contexts, have been mapped close to the mutant locus, and the two closest, or the closest ones, they were used to make sequence comparisons to Brachypodium, a related grass species. And what you can see here is that the target interval in Brachypodium comprises 6 genes that were annotated in Brachypodium, and we then went to our physical Bali map, and we were able to identify a large context that comprises those 6 genes from Brachypodium. And more important, one gene shown here in red was missing in the mutant line, and the mutant was an X-ray mutant, and therefore our hypothesis was, of course, that the mutant basically has lost the gene. Well, we sequenced, or people sequenced this back context. In addition to those 6 genes, we found some more genes here. Well, and to make a long story short, when using this or further scrutinizing this candidate gene here, we analyzed some more mutants and also a killing population, and we could confirm that a recessive or a mutation in this gene is causing the multi-noded phenotype. So this is basically a very quick way to isolate genes of interest for monogenic traits. And therefore, of course, the future will tell us, and the corresponding experiments presently are being performed, whether this kind of approach also will be useful to look at more complex genes, to look at segregating quantitative. Right. So, as was mentioned already by Swapan Datta this morning, the final goal of geneticists is to predict a genotype and to predict a phenotype. However, before we can predict a phenotype, we have to analyze the phenotype, and this brings me or takes me to the next field we are concerned with, and this is automated phenotyping. Imaging. And here at, or not here, but in our institute at IPK, we have established a few years ago a automated imaging facility having those conveyor belt system that carries about 500 parts, so it allows us to perform small genetic experiments. And one such experiment we recently have completed is the analysis of fresh weight, and in particular the question to what extent we can identify QTLs that underlie the phenotypic variation in fresh weight in a set of 100 diverse string barley genotypes or accessions. And what you can see here is that we see a fairly good correlation, more than 0.9, between the measured biomass at the end of the experiment. It was 52 days, I think 52 days after planting, and the image-based estimation of biomass. So the correlation is very good, and also the heritability of the image-based biomass is fairly high. It's more than 60%, and therefore we then took the next step and did a GWAS analysis, genome-wide association analysis, and what you can see here is the summary of the results regarding digital biomass at the end of the experiment, 58 days after planting. Well, you see there are some SNPs that clearly increase or exceed a log threshold of 3, and 2 of those SNPs here seem to be coincident with the 2 candidate genes, which we actually would expect to heavily influence biomass. The one here is earliness, photoperiod sensitivity, or early flowering, which certainly impinges on biomass development. And the next one is the benzo gene, which is a dwarfing gene, and again, we could expect that shorter plants give less biomass than taller plants. Moreover, what we see here is that there are actually the most significant QTLs are on chromosome 6, where we did not find any meaningful candidate gene meaningful to us, at least from the literature. However, surprise, surprise, what we saw here is that when we analyze for tillering, that we get the same QTL here on chromosome 6H, and that basically means that the third feature, namely the amount of tillers or the number of tillers formed by a plant, heavily influences biomass, as we could expect. And of course, it's interesting now to see what kind of genes underlie this QTL for tillering and biomass. I should also add, well, time is running, I don't add anything. So this is GWAS for biomass. Imaging is certainly a modern, high-tech approach to generate and look at phenotypic data. Nevertheless, I think as gene bankers, we should not forget that most gene banks and most curators have lots of legacy data in their files. And we really can take advantage of those legacy data. However, it is a precondition to consolidate those legacy data, because these are not clean experiments that the curators perform when they propagate the seed. We undertook such an effort to analyze these legacy data after normalizing and cleaning them. And as a matter of fact, that was quite meaningful, and that led to the discovery of novel genetic variation, again for a flowering time locus in wheat. However, things are still in process there. Now, getting back to genetics and resequencing, which is certainly something that is challenging us and has been challenging us for some time. It has been mentioned in the previous talks that genotyping by sequencing is certainly one of the bets in town that we are doing. I think it's a good exercise to get a good grouping again, there is a project underway right now to value accessions, land races, white barley, and deep PGRs using this exome capture approach that interrogates just 60 megabase pairs out of the 5.1 gigabase pairs of the barley genome. However, those 60 megabase pairs are basically holding the gene space, and therefore, from a biological point of view, it's the most significant portion right now. Well, let me conclude with a peek into the future. How can we reach out to catalog all those gene bank accessions? Now we have been just addressing 100 here, 200 there, cloning this gene and that gene. How can we put that into the context of a whole gene bank? And therefore, I want to share with you some information we put together about what is facing us when we apply this technology. And if I say what's facing us, it's basically that I'm wondering what phase the bioinformaticists who at the end of the day will use all those data. And here is just an example. So if we want to analyze the whole barley collection of IPK, that's 22,000 accessions, by GBS, 1x coverage, we generate a data volume of 6 terabytes. With exome capture, it's already 100 terabytes, and with whole genome shotgun sequencing, two-fold coverage, it's 200 terabytes. Of course, we will identify an increasing number of SNPs, but here, data analysis will take, using the established pipeline, will take 32 years for all those 22,000 accessions using exome capture, and even 64 years when doing whole genome shotgun resequencing. So I think this is something we really need to consider when planning our experiment. Some similar situation is evident when we look at phenotyping. Again, if we use our present system for image-based phenotyping, then we will generate we do generate 4 terabytes of data per year. And that's basically sufficient to analyze 800 accessions. So if we analyze all 22,000 accessions, it would generate more than 100 terabytes of data, and it would take us more than 25 years. So therefore, I think we need customized approaches. We cannot do everything in one go. We have to think about strategies that give us a staggered approach. And one of these strategies is certainly that we would start with genotyping by sequencing activity to divide our barley collection in subpopulations that we could identify. And those subpopulations will then be used, representatives of individual subpopulations, to deeper sequence, for instance, by exome capture. And finally, of course, we also have to improve on the side of informatics and bioinformatics, in particular regarding the establishment or the further refinement of a data warehouse that ties together the data from phenotyping along with the classical passport data from the genome. Well, let me conclude. You certainly agree with me that the value of a plant genetic resource is a function of its availability in a gene bank, accessibility, as well as the information on genotype and phenotype. And as I mentioned, gene banks have done an almost perfect job on seed conservation. And genomic sequences are a perfect resource for trait mapping. I hope I could show you that. And gene cloning, but the reference sequence needs to be further completed, the reference sequence of barley. Resequence and phenotyping of entire gene bank collections certainly will require adequate IT resources, and there is the need for customized approaches, because the technology continuously develops, and there is also no size-fits-all solution. So we are still in an evolving system, and we probably will need to adapt our approaches over time. And I'm leaving it there. I made it almost in time, and I'm happy to take one of the other questions.
Thank you very much. Thank you for keeping within the 30 minutes. I think we have time maybe for one or two quick questions, Roberta. Just shout. Yes.
As a director of probably the largest bank in Europe, but probably one of the largest in the world, I have a trivial but challenging question, one that I'm sure you have pondered about many times. As our genomics tools improve, more and more we move our attention to the haplotype, to the gene, rather than the whole genotype. Yet I see, particularly at meetings where different categories of people participate, stakeholders, in my opinion, there is sometimes a little bit too much emphasis on collecting, knowing that we have 70% redundancy probably in our collection. My bias is that perhaps it's like yield. If you wanna optimize yield, you have to balance the different yield components, not maximize one versus the other one. So, what is your feeling? Do you think we have collected enough and we should probably spend more money characterizing and hunting for harnessing those beneficial areas, or do you think we should collect more genotypes and perhaps send them to evolve? What is your?
Well, it's certainly a case-by-case answer, depending on the crop you consider. But as regards barley, I think we are really covering most of the diversity for the species Ordium vulgare in our gene banks. And the challenge is really to start plunging into that gene pool, because so far we really have just scratched on the surface, particular plant. So in a nutshell, we have collected probably sufficiently the cultivated, the elite germplasm, along with some of Ordium spontaneum. But in terms of further extending the collections of the genus Ordium, we definitely have to do a lot more, because in terms of the 30 white species of Ordium, just a paltry 900 or so accessions are found in gene banks worldwide.
Okay, thank you very much. I'm afraid we're running out of time for discussion as well as for analysis of the sequence. So I think we need to move on. The next talk is from Colin Kavanagh from CSIRO, who's going to talk about